So in this video we are going to discuss the details of TCA cycle and its regulation. So in previous video we have discussed that pyruvate was converted into acetyl CoA. Acetyl CoA. So what happens here? Acetyl CoA reacts with the oxaloacetate. Acetyl CoA reacts with oxaloacetate, the intermediate of TCA cycle, and form citrate, and this reaction is catalyzed by citrate synthase. This reaction is catalyzed by citrate synthase enzyme, and water enters in this reaction. Water enters and CoA leaves in this reaction. Again, the citrate is isomerized to isocitrate, and this isomerization is catalyzed by enzyme aconitase. This reaction is catalyzed by aconitase enzyme, and isocitrate gets converted into alpha ketoglutarate and this conversion is catalyzed by enzyme isocitrate dehydrogenase, and in this reaction one carbon dioxide is released, and one NAD comes and gets converted into NADH plus H. So acetyl CoA was a 2-carbon compound, and oxaloacetate was a 4-carbon compound, and citrate was formed, which was a 6-carbon compound. Isocitrate was also 6-carbon compound, but here one carbon dioxide is released, so alpha ketoglutarate is 5-carbon compound. Next, this alpha ketoglutarate is converted into succinyl CoA, and this reaction is catalyzed by alpha ketoglutarate dehydrogenase, and again here one of the carbon is released in the form of carbon dioxide, and NAD comes and gets converted into NADH plus H, and succinyl CoA is a 4-carbon compound here. Next, this succinyl CoA is converted into succinate. The succinyl CoA gets converted into succinate, and this conversion is catalyzed by enzyme succinate thiokinase, and in this reaction what happens? GDP comes, GDP plus pi, and the energy released in this reaction is used to convert it into GTP, and CoA is released here. Next, what happens? This succinate is converted into fumarate. This succinate is converted into fumarate, and this conversion is catalyzed by enzyme succinate dehydrogenase, and succinate dehydrogenase is embedded into inner mitochondrial membrane, and it is a part of electron transport chain also. And in this reaction what happens? FAD comes and gets converted into FADH2. In this reaction the energy released is not sufficient to reduce NAD, so it reduces FAD into FADH2. Next, what happens? This fumarate this fumarate combines with water. There is hydration, and it is converted into, after combining with the water, it is converted into malate, and this reaction is catalyzed by enzyme which is known as fumarase. And next, this malate is converted into oxaloacetate, the initial intermediate of TCA cycle, which was oxaloacetate. This malate is converted to oxaloacetate, and this reaction is catalyzed by enzyme malate dehydrogenase, and in this reaction again one NAD comes and gets converted into NADH plus H. Keep in mind that one of the important MCQ, conversion of malate to oxaloacetate, it is not a spontaneous reaction, it is instead an endothermic reaction. But direction of TCA cycle is always in forward, because the first reaction, which is citrate synthase reaction, uh, formation of formation of citrate from acetyl CoA and oxaloacetate. This is exothermic reaction, and this is strong enough to drive this endothermic reaction into the forward direction. So the conversion of malate to oxaloacetate is in forward direction despite of being endothermic due to exothermic nature of this reaction. So this is an important MCQ regarding the exam. So what we have studied here, acetyl-CoA reacts with oxaloacetate and gets converted to citrate. This reaction is catalyzed by citrate synthase, and this reaction molecule of water is added and CoA is released. Next, citrate is isomerized to isocitrate, and this reaction is catalyzed by a conidase enzyme. Isocitrate is converted into alpha ketoglutarate, and this reaction is catalyzed by isocitrate dehydrogenase. One carbon dioxide is released here, and one NAD is converted into NADH. Oxaloacetate was 4 carbon compound, acetyl-CoA was 2 carbon compound, citrate was 6 carbon compound, isocitrate was 6 carbon compound, and alpha ketoglutarate formed is 5 carbon compound, as one carbon is released in the form of carbon dioxide. Next, alpha ketoglutarate is converted into succinyl-CoA. It is catalyzed by alpha ketoglutarate dehydrogenase. Again, one carbon is released as CO2, and NAD is converted into NADH. Next, succinyl-CoA, it is converted into succinate. In this reaction, one GTP is converted to GTP and CoA is released, and this is catalyzed by succinate thiokinase. And Next, succinate is converted into fumarate, and in this reaction, FAD is converted to FADH2. It is catalyzed by succinate dehydrogenase. Fumarate is converted to malate by addition of water by enzyme fumarase. Next, malate is converted to oxaloacetate, and this is catalyzed by malate dehydrogenase, and NAD is converted to NADH. So these were the reaction of TCA cycle. We need to know the regulation of TCA cycle also. So now we move toward the regulation of TCA cycle. The regulation of TCA cycle is quite simple, as it does not involve any kind of hormonal regulation. So it is basically being regulated at 3 steps. It is regulated at this step. Okay, it is regulated at this step. This is first regulated reaction. Next, it is regulated at this step. This is the second regulated step, and next it is regulated at this step. This is the third regulated step. So these are the basically 3 regulated step of TCA cycle. And the first step is regulated. In the first step, reaction is catalyzed by citrate synthase enzyme, and the product of this reaction is citrate. So basically this step is inhibited. This is inhibited by its product. So in this step, the product of reaction is citrate, and this citrate inhibits citrate synthase enzyme. So in this way, the step is regulated. Moreover, the availability of substrate also influences the first reaction. If substrate is surplus, the rate of reaction will be greater. If substrate is less, the rate of reaction will be less. And next, it is regulated at this step, the conversion of isocitrate to alpha ketoglutarate, and it is regulated by energy signal. High energy signal such as ATP and NADH inhibit this reaction and decrease the rate of TCA cycle, while low energy signal such as ADP stimulates this reaction. Similarly, calcium also stimulates in this reaction. Calcium is released during skeletal muscle contraction. During skeletal muscle contraction, there is elevated level of calcium in the cell, and this elevated level of cell stimulates this reaction in the forward direction also. And next, the third step of this reaction, which is alpha ketoglutarate to succinyl-CoA, it is also regulated in response to the energy signals and calcium. Calcium is basically stimulator of this reaction again, and the high energy signal such as NADH is inhibitor of this reaction. And moreover, this reaction is also subjected to product inhibition. The product of this reaction is succinyl-CoA. This succinyl-CoA also inhibits alpha ketoglutarate dehydrogenase enzyme. So it is basically stimulated by calcium and inhibited by NADH and succinyl-CoA. So this is the regulation at 3 steps of TCA cycle, and the rate limiting reaction is this one, as this is the slowest reaction of this pathway. It is rate limiting reaction of this pathway. And one more thing here, the enzyme aconitase is also it is viable, or it can be inhibited by rat poison. Okay, rat poison contains the compound which is called fluoroacetate. This fluoroacetate, this fluoroacetate gets converted into fluoroacetyl CoA, and this fluoroacetyl CoA combines with oxaloacetate. This fluoroacetyl CoA combines with oxaloacetate, and after combining with oxaloacetate, it produces uh, fluorocitrate. After combining with fluoroacetyl CoA, it produces fluorocitrate, and this fluorocitrate is inhibitor of aconitase enzyme. So rat poison can inhibit the aconitase enzyme. So it was the regulation of TCA cycle and reaction of TCA cycle. In the next video, we will discuss the energy release in TCA cycle. Thank you.